Please take your seats and welcome the President of the American Epilepsy Society, Dr. Howard Goodkin.
Welcome, everybody, and thank you for joining us this afternoon. I'm delighted to welcome you to the 23rd Judith Hoyer Lecture in Epilepsy, Treating Seizures, Patterns, Past, Present, and Future. The Judith Hoyer Lecture in Epilepsy combines the discussion of the state-of-the-art in a specific area of epilepsy and the impact it may have on patient care and outcomes. We are proud to partner with the National Institute of Neurological Disorders and Stroke in presenting this annual session. We are also pleased to continue to make this lecture accessible to both professional and public audiences. For the 7th year, we are streaming live webcast of this public lecture, so many more listen live or the archived recording afterwards. I want to acknowledge the support and educational grant from Upshur Smith and the support of the live stream of this lecture by Jazz Pharmaceuticals. At this time, I want to recognize the companies that support AES through medical education grants, charitable grants, sponsorships, exhibiting, and advertising. I invite the representatives from these companies to stand as I read your company's name. At the very highest level of support, thank you to our benefactor level supporter, UCB Incorporated. Thank you to our leader level supporter, Jazz Pharmaceuticals Incorporated. Thank you to our partner level supporters. Thank you to our partner level supporters, BioCodex Incorporated, Biohaven Pharmaceuticals Incorporated, H. Lundbach, AES, LivaNova, Neurelis Incorporated, NeuroPace Incorporated, Praxis Precision Medicine, SK Life Sciences Incorporated, Stoke Therapeutics Incorporated, and Xenon Pharmaceuticals. Thank you to Acadia Pharmaceuticals Incorporated, Octa Pharmaceuticals Incorporated, and Children's Health for participating at the supporter level this year. And lastly, to Med Learning Group, Neurona Therapeutics, Persist Development Corporation, and Upshur Smith Laboratories, LLC, for participating at the contributor level this year. Now I'm very excited to present the Distinguished Service Award, which was established in 1993 and recognizes outstanding service by an AES member in the field of epilepsy with special emphasis on exemplary contributions in service to the AES, its members, and our patients. I am most pleased to present the 2025 AES Distinguished Service Award to Dr. Farah Lubin. I have to still introduce you. All right, I'm going to make Farah stand here as I read all the wonderful things about her. Dr. Farah Lubin is a distinguished and Triton-endowed professor of neurobiology at the University of Alabama, Birmingham. She earned her BS summa cum laude in biology and chemistry from Alabama State University, her PhD in cell molecular biology from SUNY Binghamton University, and completed 2 postdoctoral fellowships at Baylor College of Medicine. Dr. Lubin is director of the NIH-NINDS Neuroscience Roadmap Scholars Program at UAB, which aims to enhance engagement and retention of graduate trainees in neuroscience. Dr. Lubin is focused on studying the epigenetic basis for transcription programs in the healthy and diseased brain. She and others have observed that neurons have hijacked epigenetic processes such as DNA methylation, post-translational histone modifications, and non-coding RNAs to coordinate gene transcription changes in mature, non-dividing neurons during memory formation. Dr. Lubin uses interdisciplinary methods ranging from molecular neuroscience to cutting-edge tools such as CRISPR-Cas9 gene editing to understand how neuronal activity alters the epigenome to direct gene expression patterns. Her work has provided insights into epigenetic mechanisms that participate in the regulation of gene expression during memory encoding, allocation, storage, and recall in hopes of unraveling the causes of cognitive deficits and to develop treatment options. Dr. Lubin, you're already here on the podium with me, and you are well deserving of this honor. And so with my heartfelt thanks for your extraordinary service.
Thank you, Howard. Can you guys hear me? Hello.
There you go.
Good afternoon, everyone. I wrote a very short thank you speech, so please give me a minute, and I'll try not to be too emotional. I am deeply honored to receive the 2025 AES Distinguished Service Award. This recognition means so much to me because it reflects values I hold dear: service, mentorship, and advancing the epilepsy field. When I joined AES over 20 years ago, I could not have imagined the incredible journey ahead. From serving on AES committees and the Epilepsy Currents editorial board to chairing the Diversity, Equity, and Inclusion Committee, my goal has always been to strengthen our community and create opportunities for others. I want to share a brief story from my early career. When I first entered neuroscience, I had a PhD in immunology and no background in epilepsy research. I vividly remember walking into my first AES meeting feeling both excited and overwhelmed. What made the difference were the mentors and colleagues who welcomed me. For example, Dr. Manisha Patel, who will be speaking with us soon. They were able to answer all of my questions and encouraged me to keep going. That experience taught me the power of community and the importance of lifting others as we climb. It's why I've dedicated so much of my career to mentoring and helping to create programs like the AES Bridge program, which helps underrepresented students find their place in neuroscience. This award is also very personal. As a young Black girl growing up in New York City, becoming a doctor seemed like an impossible dream. Thanks to mentors, outreach programs, and scholarships, I found my path. I have dedicated my career to ensuring others have the same chance. I want to thank AES for this honor, my colleagues for their collaboration, and my trainees for inspiring me every day. I promise to continue working to advance epilepsy research, promote diversity, and support the next generation of scientists. Thank you.
All right, he's walking away. Oh, can we get one? Gosh, there's it.
Thank you.
Thank you. Well, I get to follow one exciting award and well-deserving award with another. So it's now my distinct honor to present this year's Extraordinary Contributions in the Field of Epilepsy Award to Rosemarie Cabal. Rosemarie worked with the Center for Disease Control and Prevention Epilepsy Program for 24 of her 25 years at the CDC. She has 2 master's degrees, one from Emory University Rollins School of Public Health and from the University of Pennsylvania. Following her work at the Carter Center, she began her CDC career as a research fellow and within a year was hired to work with the CDC Epilepsy Program. She initiated epilepsy surveillance on a national and state public health surveillance system and led population studies of epilepsy prevalence and its corresponding burden. Rosemarie led the development of the CDC Managing Epilepsy Well Network that has contributed to closing epilepsy self-management research and program gaps, and that became a model for other CDC research networks. Between 2012 and 2015, she served as the epilepsy program team lead, effectively guiding program activities consistent with the IOM recommendations to CDC and with agency priority areas. She has published over 80 articles and government reports, including 63 focused on epilepsy burden and disparities. She has served multiple agency emergency responses and has led multiple federal committees that set national objectives to improve population health, including for people with epilepsy. Rosemary, please join me at the podium and accept this well-deserved award.
Good afternoon, everyone. Thank you, Dr. Goodkin and AES board. I am immensely honored to receive this award from the American Epilepsy Society. I couldn't begin to tell you how personally meaningful it is to me in just the few minutes that we have. And yet, although I am absolutely grateful for the individual recognition, I do want to share it with my former CDC colleagues and our awardees who have worked very hard over the years implementing our public health initiatives across the country. These initiatives reflect partnerships and community engagement and community interventions necessary to improve the conditions for people with epilepsy, above and beyond the treatment and care and the dedication that you provide to your patients. And that, in a nutshell, is the essence of public health, the assurance of the conditions in which people can be healthy. To say that we're living in contentious and tumultuous times for public health, specifically, and society here in the US in general, is an understatement. But for those that question investment in public health action on epilepsy, it's important to remind them loud and clear, and as my own immigrant parents, my Italian immigrant parents would say in Italian, chiaro e tondo, loud and clear, that through our collective efforts, we have moved the needle over the years. We did move the needle on improving visits to neurologists for adults with uncontrolled seizures throughout the previous decade. We did move the needle on improving health insurance coverage for people with epilepsy after implementation of the Affordable Care Act, dare I say Obamacare, now that I'm no longer a federal employee. I can say those words now. We did move the needle on improving some public knowledge and attitudes about epilepsy, and that needle is shifting in the number of adults certified in seizure first aid in both English and Spanish, creating that more supportive environment for people with epilepsy. And that needle is shifting in the reach of evidence-based epilepsy self-management programs in local communities to help people with epilepsy with their self-care. And we also showed where the needle is moving in the wrong direction around some risky behaviors like preventable cigarette smoking and higher mortality rates for people with epilepsy, in particular African Americans and men. And within the midst of the COVID pandemic, we tracked COVID vaccination status, COVID testing, and emergency department use among people with epilepsy to assess their status and to see if we needed to redirect any resources to support our population. These and so many other outcomes exemplify the success that is possible through organized community effort, and they underscore the urgent need for continued public health capacity for epilepsy. I've been both exceptionally privileged and proud to work with so many amazing colleagues at CDC and elsewhere, and with amazing epilepsy researchers and leaders and clinicians, such as all of you here, to support our public health action on epilepsy. And I certainly look forward to seeing our continued progress in the decades ahead. Thank you so much.
Thank you, Rosemary, and congratulations to both you and Farah. Again, I've said it a couple of times, but both very well deserving of these awards. Core to the AES mission is the development of the next generation of basic translational and clinical investigators. These young investigators are indeed the future of our community, and they are a vital part of our society. AES encourages young investigators to attend the annual meeting by providing travel and other awards in recognition of the most outstanding abstract submissions. You can see the listing of these early career investigators in the program book, I think I mean the app, and I encourage you to visit their posters. In addition, AES supports research through more than $1.5 million in fellowship grants and awards. An example of a partnership to ensure funding for early career investigators is the AES, American Brain Foundation, AAN, Epilepsy Foundation, Susan S. Spencer Clinical Research Training Scholarship in Epilepsy. It is my pleasure to introduce Dr. Peter Hayter from MGH Center for Neurotechnology and Neurorecovery. His project focuses on developing a virtual neurostimulation model for temporal lobe epilepsy therapies. Please join me in congratulating Dr. Hayter on this prestigious award. The Susan S. Spencer Clinical Research Fellowship is made possible by the generous personal gifts of AES members and others who have made contributions to the endowment that supports this fellowship. It only takes a minute to support the Susan S. Spencer Fund, the Lennox and Lombroso Fund, or the Sergevsky Fund. Visit the AES website to make a donation to help AES in supporting the career pathways of the next generation of researchers. AES has made more of the seahorse pins that I'm nicely displaying up here that many of you have asked about. You cannot buy these pins anywhere, but you can get one as a thank you if you make a pledge of $1,000 per year for 3 years to the Susan S. Spencer Fund. Please stop by the AES Service Information Desk near registration if you want to pledge and take home a pin. Thank you all in advance who will stop by the service desk to make that decision, that the donation, have already made your donation, or will do it when you get home. Again, it's for a good cause to support the next generation of epilepsy researchers. The AES is also very proud to be the administrative home, as well as a member of the Epilepsy Leadership Council, a coalition of nonprofit professional societies, patient advocacy organizations, and federal agencies, all working together to expand our impact and improve the lives of individuals living with epilepsy. And now I'd like to invite Laura Widener, Chair of the ELC Steering Committee, to come to the podium.
Hello, everyone. Dr. Goodkin, thank you so much for the opportunity as current chair of the Epilepsy Leadership Council to share some brief thoughts. As many of you know, the ELC was formed in response to the 2012 IOM report on the epilepsies, which called for an expansion of the Vision 2020 collaborative efforts. More than 10 years later, ELC is more than 70 members strong and unites the epilepsy community to address gaps in support, education, clinical care, and research. By sharing information, resources, and opportunities, the ELC is one of the key coalitions that facilitates collaboration on shared goals and increases the community's impact for people with epilepsy and their families. A saying that I live and work by is, we can do anything, but we cannot do everything. Each of us and each of our organizations brings unique perspective, experience, and skill, but only by banding together whenever we can will our voice be louder and our strength be greater. Uphill battles surround us, perhaps a common theme of these times, but I'm so proud to be standing alongside each of you and of the progress we are making together. The slides you saw when you entered the hall or logged into the live stream showed the work of many ELC member organizations, many of whom are exhibiting in the Epilepsy Resource Center. I encourage you to visit the booths and hear more about the incredible work that they are doing for the epilepsy community. I would also like to thank the American Epilepsy Society for hosting the ELC and providing leadership, coordination, and support to ensure our collaborative work continues to move forward. Please stop me in the hallway or grab one of the AES staff to learn more about the ELC and be part of our amazing collaborative community. Thank you very much.
Thank you, Laura, and to the steering committee for your leadership of ELC. I'm now pleased to introduce Dr. Walter Koroshet, Director of the NINDS, and one of my early attendings at the Mass General. Dr. Koroshet's presence reflects both the long history of strong NINDS support for the epilepsy community, as well as his personal commitment to epilepsy research, and we sincerely appreciate his taking his time from his busy schedule to be here with us again this year. Thank you.
It's not that busy. But it is a joy to be able to come here and talk to you a little bit about what's going on at NINDS and a couple of tidbits about what everyone else here is doing with the funding from NINDS. So again, I'm always a pleasure to introduce the Hoyer Lecture. It's named after Judith Hoyer, the wife of Representative Congressman Steny Hoyer of Maryland. She was a member of the Epilepsy Foundation Board. She was dedicated to improving the care of people with epilepsy and their quality of life, as well as education of children. Unfortunately, she passed. She did suffer from epilepsy, and you'll be hearing from her husband, the congressman, at the end of this talk. So lots of changes have happened, and I'm going to talk to you a little bit about the changes, but I think the main thing to remember is that NINDS is focused, has not changed. Our focus is on supporting the best science in both the basic fundamental science, translational science, which is development of therapies, and clinical neuroscience, understanding from patients, trying to understand from patients better ways of treating epilepsy and their disorders that cause them, and that's really not going to change. We also are committed to training the next generation of scientists. That's really been the secret to our success, that we put a lot of time and effort and money into ensuring that people who want to spend careers in research have the opportunity to do so. And that's really so important. As you'll hear in a few slides, the future is just getting more and more exciting as the new tools are coming out. And then to learn from you, the epilepsy community, and to try to disseminate the knowledge that's coming out of the NINDS research is also part of our mission. Now, the key thing about the NINDS is the core is getting the money that Congress appropriates to us out to the best science. And lots of things have happened in the last year, but our people worked really, really hard. They worked overtime, they worked weekends, and they got every penny out by the end of the year, which for NIH is September 30th. And so we have a really dedicated staff. We don't have a final budget yet. We're operating under a continuing resolution, which means operating as if we had the same amount of money that we had last year. But the House and the Senate did put a bill together for NIH, which is pretty much a flat budget, maybe a slight increase for NINDS. And you can see here, the base budget is the budget that's available for everything. And then we have congressionally mandated programs. The BRAIN initiative, as I mentioned before, which I've mentioned each year, is so exciting for epilepsy because it's really trying to understand how to monitor, map, and modulate circuits, which is critical to epilepsy research. The HEAL initiative is to develop non-addictive pain therapeutics. The Undiagnosed Disease Network we run, and that's for mostly kids who have diseases that were never diagnosed. Most of them turn out to be, about 80% have a neurologic component. And then we have some money for a particular interesting program in ALS, and there's a new bill called the National Parkinson's Project, which doesn't have funding for it. For research now, has some money to kind of organize a plan for Parkinson's research. So that's kind of what it looks like. We have to wait and see if the Congress can pass a bill for all this to happen. But as I said, right now, we're operating under continuing resolution, which is which is, in both cases, we're pretty much going to have a flat budget. Now, some things have changed, and so the people who are involved in NINDS funding, these are the kind of core things to know. We used to review things, some grants in our institute, that's gone. All applications are now reviewed in the Center for Scientific Review. Beginning in FY 2026, all the information about grants will be in grants.gov. Currently, it's on the different NIH funding pages. There will be a decrease in the number of what we used to be called RFAs, then termed NOFOs. They've been asked to reduce these by about half. Some of these will be replaced by something called highlighted topics, which are an institute's notice to the public that they're interested in this area of research. So highlighted topics and NOFOs will still exist, and those are the kind of things to track to see if your research fits into one of those, because they will have some increased propensity to be funded. There's a real emphasis in the government now to be more stringent about funding overseas, so there's going to be special processes that are going to be required for any grants that have an overseas component. NIH is also prohibiting the institutes from acting on a strict pay line. So NINDS, we would establish a pay line. If you got below that percentile score, you pretty much guaranteed to get funding. That's gone away. We still, will basically rely on the peer review as the most important factor, but there will be some, what we call skips and reaches. We used to have these, we put them in a bundle called high program priority projects, but I think for us, the general funding strategy will be very similar, but we won't have a pay line, so it will be unclear for an investigator when he gets a score to know for sure whether he's going to be funded or not, because we won't be able to announce that pay line every year. And then we will continue to support the trainees, as I mentioned before, that's so important, and we will continue to try to spread the funds as best we can, and this came from the last couple of years, because with the flat funding that we've had, the cost of grants went up, so we could fund fewer grants. So what we have done is we've made it really much harder to get a grant if you already have $1.5 million total cost from NIH, or if your grant costs more than $500,000 per year. So those will continue going into the future until we get some relief in terms of the budget, a budget increase that could then match the inflation. So that's the new things. We still have our website page that'll probably go away in a year, and there'll be a consolidated NIH website page. The program offices are really more important than ever now because things are changing quickly. I don't even know what they are all the time. Program offices are often the first to know, and they can be incredibly helpful. And then again, by 2026, have to get familiar with grants.gov, which is the website for all government grants, but you basically check HHS, and then you check NIH, and then you'll get into the NIH section. Okay, so I just wanted to go then to talk a little bit about the epilepsy research at NIH. So you can see here the funding, it went up during the period of time where we got increases, and now it's flat when the budget got flat. And about 80% of the epilepsy projects at NIH are funded by NINDS. The epilepsy screening program, I hope people know about this. It's actually in its 50th year. It's a program where people can come in and for free have their agents tested in multiple different models of epilepsy. It's run by Karen Wilcox out at the University of Utah, and multiple different drugs that have gone to the market have gone through this program, and there's a number that are in the program now that we expect to come out onto the market too, hopefully. We have some very interesting Center Without Walls projects where we encourage people to form a team, attack a problem, come up with really important solutions to some of these problems. And these have been very successful, and we've actually used this to put together other programs, what's called the RM1 program, which is a team science program at NIH, but it's really based on the success of the CWOWs. In status epilepticus now, we actually have grants going on in the pre-hospital space, the emergency department, and the in-hospital setting now. We have a number of rare disease clinical research networks that are looking at conditions that are associated with usually severe epilepsy. And, you know, if I have one beef to get out to people, I think that it's really important for the society and the people in epilepsy to take ownership of the disorders, the developmental disorders that cause these terrible seizures. Because you're treating the patient, not just their seizures, you're treating the patient as a whole. And these disorders are now going to become treatable with some of the new genetic therapies. Some advances to mention from the SUDEP, CWOW, really interesting data on risks that might be associated with SUDEP, both in terms of periictal apnea and differences in altered slow wave activity progression during non-REM sleep and interbreath intervals during non-REM sleep. So this has been an incredibly productive group, and I think helping to understand the risks that hopefully we can then translate into some kind of precision prevention. Really interesting stuff going on now in the gene therapy space, and this is a really interesting paper. It's basically riffed off of the Brain Initiative, which is heavily run out of the Allen Brain Institute with Ed Lean. And in this group, they developed a very clever way of getting the SCN1A gene into the mouse in a mouse model of Dravet syndrome. And the interesting thing about it is that if you just give the vector with the gene, the mice don't do well at all. But if you target just the GABAergic neurons, then you block a lot of the seizures and you improve the animal's behaviors. So, and you can do that now because with the Brain Initiative, people find enhancers and promoters that are cell type specific. So if you want to alter some gene, you can actually alter it just in the cells that you want to alter it, not in the entire brain, which is related to the fact that drugs are dumb. They don't know where to go. But with these therapies, these are really smart therapies. And lastly, I think there's a study which I found very interesting using stimulation to kind of block the network connectivity that occurs in the interictal period as a way of potentially treating epilepsy. So we focus so much on preventing the seizures when the seizures occur, but a lot of stuff is going on in the brain. Every time you have a spike or an epileptiform discharge, even though it's asymptomatic, it's affecting the circuits. And now, people are trying to get in and affecting that network itself, not necessarily just the seizure, but that may have really strong effect sizes. There's another example of an interesting study where they use knockout for DEPD-C5, which is in the mTOR pathway. And the important thing here is that if you knock out you make a knockout mouse, then they have problems with organization of the cortex, and they have seizures. In this experiment, they basically do the knockout after the animals are born already, so the actual cortical migration is normal. They still get a lot of the features that you see in like TSC, but it takes a long time, and it occurs as the mice get older, and then it gets the seizures. So the point is that you don't need actually the cortical development abnormalities to get the seizures and a lot of the functional stuff. That this is occurring even in the postnatal space. And lastly to say, NINDS is our 75th anniversary. I'm really excited that the AES has really come forward and helped publicize that. I want to thank all our staff, many of which are here, who've really worked incredibly hard through tough times to really help facilitate your science and get it done for the patients with epilepsy. And lastly, to say that it's just a joy to mention and announce that Dr. Manisha Patel is going to give the Hoyer Lecture in a couple of minutes. She's the Professor and Associate Dean of Research at the University of Colorado. She's had 25 years of continuous NIH funding, including the Javits Award, which is the NINDS Lifetime Achievement Award, which you don't have to be really old to get. It just means you did a lot of good stuff really quick. And she's also the former AES president. So with that, I want to hand over to the video from Congressman Hoyer. Thank you.
Hello, I'm Steny Hoyer, Congressman from Maryland's 5th District and a former Majority Leader of the U.S. House of Representatives. Welcome to the American Epilepsy Society's 2025 annual meeting. You're all here because you recognize the challenges that came with an epilepsy diagnosis. That's something my family certainly understands. My late wife, Judy, lived with epilepsy. Crucially, you came together today because you understand just how vital it is that we improve our ability to understand, treat, and cure epilepsy in all of its forms. The AES annual meeting is a key part of that effort. Whether you come from academia, clinical practice, private industry, or the advocacy community, thank you for making this gathering a success and for your work on behalf of the epilepsy community. That includes Dr. Walter Koroshetz, who continues to lead the National Institutes of Neurological Disorders and Stroke with great ability and vision. I especially want to thank the numerous people living with epilepsy who share their stories, not only at the AES annual meeting, but throughout the year. As a legislator, I know how powerful it is when a community can build consensus and speak with one voice. That is what all of you have done. I know that I'm working to build that same consensus on Capitol Hill, alongside the rest of the Congressional Epilepsy Caucus. That's what I've worked to do ever since partnering with my dear friend Tony Coelho to bring Democrats and Republicans together to pass the Americans with Disabilities Act 35 years ago. We need to foster that spirit of solidarity today amid this challenging moment for the epilepsy community, indeed, for all America. That means pushing back against partisan attacks on Medicaid and Affordable Care Act subsidies that have led millions of Americans, including many with epilepsy, to lose access to life-saving care. That means defending the CDC epilepsy program, the BRAIN Initiative, and other federal research initiatives established and funded by law. And that means ensuring the Department of Justice and the Department of Education fulfill their obligation to protect the rights of Americans with disabilities, including, of course, epilepsy. We can't be just reactive, however. We need to be proactive. That's why I'm proud to support the National Plan for Epilepsy Act. That bipartisan, bicameral legislation already has support from more than 40 members of Congress and 150 epilepsy organizations. I am hopeful that it would bring much needed attention, resources, and support to our work on epilepsy. But we need to get this legislation across the finish line. That will take each one of us, whether you're a doctor or a nurse, a researcher or an advocate, a member of Congress or a patient, working together to build consensus. That's what Judy would tell us to do. She would be so proud to see all of you coming together in common cause. People like our speaker for this year's Judith P. Hoyer Lecture in Epilepsy, Dr. Manisha Patel. Dr. Patel is a professor of pharmaceutical sciences and associate dean for research and graduate studies at the Skaggs School of Pharmacy and Pharmaceutical Sciences at the University of Colorado Medical Campus. She's past president of AES and has received over 25 years of continuous NIH funding to support her lab's work. Her research program seeks to understand the redox and metabolic basis of epilepsy and develop metabolism-based therapies for its treatment. Her work has provided compelling new directions for treatment development in some of the most challenging epilepsy syndromes. I hope what you learned from today's presentation will fuel your vital work on behalf of the countless people who, like Judy, lived with epilepsy. Thank you. Thank you for all that you do for millions of people. Godspeed.
Thank you. Thank you so much. It's just an honor and a privilege to be able to deliver the Judith Hoyer Lecture. Dr. Koroshets, thank you for your kind introduction, and Dr. Representative Steny Hoyer for the kind introduction. I'm going to start out with my disclosures and acknowledgments. I don't have any conflicts to declare, but I'm so very grateful to the funding from NINDS that has really fueled my research. So I started my NIH career, I would say, in academia through receiving a T32 slot in the Duke University cerebrovascular disease area, and Dr. Jim McNamara, past president, and I went to his lab and I studied the basic mechanisms of glutamate excitotoxicity. And 25 years later, I'm so proud to receive the Javits Award. But going from a T32 funded award to the Javits Award is not an easy path, and I really want to thank some of the other agencies, including the AES, for funding some of the small projects, and the Epilepsy Foundation, CURE, and Dravet Syndrome Foundation, because without that sort of seed funding, it is not possible to get NINDS funding. So thank you. I also want to thank all the trainees, students and postdocs, and my collaborators and colleagues for their help with the research. So the learning objectives are simple. How does energy metabolism get altered in epilepsy, and be able to describe how metabolic functions and redox pathways can get disrupted in acquired models of epilepsy as well as genetic models, and state some metabolism-based therapeutic targets for epilepsy. So my talk is really divided into 4 areas. I want to take you through a metabolic fuel journey and give you a basic plan for how the brain works in terms of energy, and then give you 2 examples, one in a genetic syndrome and then acquired syndrome, with some experimental data from humans and animal models and therapeutic approaches, both pharmacological as well as ketogenic diets. So let's start with the very beginning, which is what is the meaning of life, basically. And I know you've asked this question to yourself, or someone has asked you this question, and let me give you the answer from an energy basis, which is that it is simply harnessing the energy from plants and using the oxygen from, harnessing the energy from food and the oxygen from plants, which feed into the mitochondria. And in the mitochondria, there is a reunification of these electrons from these carbon sources with oxygen that we have opted to be our final electron donor. And we get this proton pumping and the energy charge, which one would call the holy grail of life, that gives us ATP to sustain the energy for our brains and warmth for our bodies. So the energy to our brain, it really transforms into everything ranging from emotions to our health in disease states and in normal, and it takes you through all the brain functions. So why does this brain need this sort of energy? And what really is the metabolic stress that the brain experiences is due to the disproportionate amount of energy that it needs, the oxygen that it needs on its weight basis. So for its 2% weight of the total body, it needs 10 times more energy in the form of oxygen. And this is the greed for oxygen and energy to sustain all the cellular processes and brain functions that are important for the brain to work. And these include postsynaptic transmission, neurotransmitter biosynthesis, action potential, the resting membrane potential, and all of the cellular machinery of the brain. So if, in terms of controlling seizures from a metabolic standpoint, we've really been amiss in terms of just looking at the tip of the iceberg, if you will. So when you look at the base, we've sort of tried to control or rebalance the excitation inhibition imbalance, but when you look underneath, there's a whole sea of metabolism which can be controlled through fuels and metabolites. And so I want to take you through some of those processes. The question I want to raise, which is the title of my talk, is there a metabolic paradigm for epilepsy and its comorbidities? And this comes from lessons from neurodegeneration, conditions like Alzheimer's disease, Huntington's, Parkinson's, and ALS, where a shared metabolic paradigm does exist. And it is based on a progressive observation of glucose hypometabolism that's noted in most of these conditions, and defects in mitochondrial and energy processes that are in some brain areas that are vulnerable. So what about the epilepsies? Well, compared to the neurodegenerative diseases, the epilepsies tend to be very complex. They range from pediatric to adult conditions. They're very unique in their circuit-based versus brain region type of condition. There are phases of metabolism during ictal phases and hypometabolism during interictal phases, but an overall picture when we measure is a hypometabolic phenotype. And then we have neuronal damage, which is not uniformly prevalent in all of the different disorders of epilepsy. However, we have observed inflammation in many conditions as well as mitochondrial defects. So the question is this sufficiently explored in neurodegenerative diseases? And the answer is yes. However, when it comes to the epilepsies, this question is still open. And I want to take you through some examples to show you that there may be a precedence to consider this metabolic paradigm. Another way to view this is through the metabolic iceberg. And this is a hypothesis that is put forward by Phillips and Picard for the neurodegenerative diseases. And what it does is it goes through, commences at the base with some of the key abnormalities at the cellular and metabolic levels that may occur in these diseases. And this may include genetic changes, it may be abnormal protein aggregates and different types of environmental insults in conditions like Alzheimer's or Parkinson's disease, and then proceed through the bulk of the iceberg where you can see the metabolic impairments in mitochondria and other processes, into the tip where you see the symptoms of the disease, where depending on what neurodegenerative disease you have different types of hallmarks and symptoms. So if we were to put epilepsy in this iceberg framework, you would have the genetic, metabolic, structural, infectious types of etiologies at the base. And through the bulk, you may see some hypometabolism and impaired energetics depending on the type of epilepsy that's been examined. And at the tip of the iceberg, we may see the core symptoms, the seizures, comorbidities, and at a physiological levels, an excitation inhibition imbalance. So the question that is raised, is there a metabolic paradigm in epilepsy and or its comorbidities, really has to do with finding the evidence for metabolic alterations that may be common, underlying mechanisms, the basis of some of the clinical features and anatomical pathological features, and most importantly, response to therapeutic interventions. And one of the questions we can start with is what is the most notable metabolic alteration that is common to neurodegenerative diseases and epilepsy? And that, I would argue, is hypometabolism that is observed in epilepsy as well as some of the chronic neurodegenerative diseases. So on the left top, you can see Alzheimer's disease brains by PET imaging, and we also see hypometabolic phenotypes in the temporal lobe epilepsy in the human condition, as well as in animal models of temporal lobe epilepsy. And at the bottom, there is a brain of the Parkinson's disease patient showing the similar alterations. So there's reduced glucose utilization during the early stages of many of these neurodegenerative diseases. And now when you put the framework of how energy metabolism can impact epilepsy and importantly its comorbidities, and if we were to consider this 10 times the energy utilization of the brain, but make it finite, so we give an energy budget to the brain, which is fixed and finite, then you can kind of plug in all the processes that occur in the epileptic brain, such as the changes in EEG, the ictal changes that correlate with hypermetabolism, the interictal changes that correlate with hypometabolism, and postictal changes that may have a combination and difficult to measure by imaging modalities. There's a lot of cost, energetic cost to all of these processes that can increase the activation of signaling pathways or transition from one state to the other, inflammatory conditions. All of these are energy utilizing processes. So the costs will have a real impact on and consequence to the brain functions. This includes alterations in excitability as well as a diversion of these finite resources, this fixed budget, to conditions that may need them more, such as proper brain function, cognition, and learning and memory. The adaptive responses of the brain can also be impaired because of these costs. And then most importantly, sleep, which can be a comorbidity, and aging process itself can be accelerated. So now we can kind of put down this framework and imagine how it would impact the epileptic brain when you have these energy constraints and impositions on our energy budget. So I'm going to give you a very short Biochem 101, starting with what we use as energy sources, and it's simple. It's glucose. Glucose is our primary energy fuel for the brain. It's the preferred fuel. And normally, glucose oxidation is very efficient. It feeds into the TCA cycle and then into the mitochondrial electron transport chain to generate ATP. The most important part of this slide is that glucose oxidation and oxidative phosphorylation are typically matched 1-to-1. So there is no mismatch that occurs. However, when you look at the epileptic brain, when neuronal excitability is increased, and this is a slide from the paper of Gary Yellen, who has very elegantly shown that when neuronal excitability is increased, there is an increase in neuronal glycolysis. And this is sort of heretical if you think about it, because you would expect the brain, the neuron, to use mitochondrial oxidative phosphorylation, but instead it chooses to increase glucose locally in the neuron. And this has also been observed in cancer cells in a phenomenon that's known as the Warburg effect, in which oxygen, despite its presence, there is an increase in glycolytic flux. And this results in the production of lactate, which is a metabolite that can be utilized to regenerate NAD to kind of balance things. But what is important about this phenomenon is that you increase glycolysis but not oxygen consumption, and this is really an odd phenomenon in metabolism. So what are our alternative fuels that we could use in the brain? One such fuel, in addition to glucose, is glycogen. So it's a storage for glucose. Lactate, as I mentioned, could be used, ketones and fatty acids. But what's interesting is glucose is like liquid cash. We all prefer to use that. Glycogen would be like a checking account. It would be there when we need it. And ketones and fatty acids are somewhat longer term savings accounts, or maybe if you buy gold from Costco and you keep it there and you think you're going to use it at some point. Hopefully we don't have to adapt to that, but we often do. We switch to using glucose and fatty acids in hard times. So now we have these alternate fuels that could be really harnessed to give us good therapeutic entities when we need energy in the epileptic brain. So with that sort of short lesson, I want to take us through examples of metabolic alterations in a genetic epilepsy situation. And this is a slide from Jung Ro and Detlef Boisson's very elegant article, and it has to do with the pure metabolic epilepsies. So the pure metabolic epilepsies are complex disorders in which we see neurodevelopmental alterations, cognitive changes, as well as seizures. And they may arise due to inherited or congenital causes where epilepsy occurs due to alterations in substrate, transport, neurotransmitter metabolism, alteration in the astrocyte coupling, or a receptor function itself. And they all kind of impinge on mitochondrial energy production, so there's a decline in that and an increase in the production of reactive oxygen species. But what about a genetic disorder such as developmental and epileptic encephalopathy? And one such example is Dravet syndrome, which was just brought up by Dr. Koroshetz. It's a prototypical genetic epilepsy in which there is mutations that could be truncation or missense mutations for the most part in the sodium channel SCN1A that encodes for NAV 1.1. And these children exhibit febrile seizures typically before age 1 year of age, and progress to very severe and spontaneous seizures. They have many comorbidities and developmental delays, which are just as disturbing as the seizures themselves, and may not respond to all the medications that are typically presented to patients with epilepsy. Interestingly, they do respond to ketogenic diets, but also have a very high rate of unexpected death due to epilepsy. So one of the studies that is done in human patients, in a small number of human patients, these were genetically verified for SCN1A mutations, and this study showed an age-dependent decrease in glucose hypometabolism. So children did not experience this change early on, but as they aged from the years 2 years to 29 years, they showed a progressive decline in glucose consumption by this FDG PET analysis. So this kind of shows you that and this is just one of the studies that was done, that glucose hypometabolism may be a key factor in humans. What about further evidence in patients with DS? Well, patients with DS, as I mentioned, are responsive. They achieve greater than 50% reduction in their seizures when put on the ketogenic diet. And there has been at least 2 studies done in peripheral tissue in muscle biopsies from patients with DS. They have shown some mitochondrial electron transport chain defects, and skin fibroblasts from children with DS have also shown reduction in their ability to form energy through oxidative phosphorylation. However, these are invasive methods for understanding the mechanistic basis of metabolic impairment in these patients. So the question is that we raised was, can peripheral cells or tissues show metabolic defects in these neurological disease and in conditions like DS? And there is some precedence for this that comes from other disorders of the nervous system. For example, in Parkinson's disease, it was shown in the 1980s that platelets from blood had a complex 1 dysfunction in mitochondria, and this was then validated in the substantia nigra pars compacta from postmortem brain tissue in patients. In autism, lymphoblast cell lines, which I'm going to tell you a little more about, which are immune cells from the blood, have shown mitochondrial defects, and that's a neurodevelopmental disorder. And then in schizophrenia, more recently, this interesting study which compared the lymphoblast cell lines with neurons that are derived from induced pluripotent stem cells, and these show similar defects in their mitochondrial energy production, which is interesting. So this gives us a precedent to ask, can we develop a cell model from these cell lines which we can compare controls and patient cells to ask if there is any metabolic defects. And this work was done in collaboration with Dr. Kelly Knupp in Children's Colorado and conducted by Anna Figueroa, who is a PhD student and also holds a PharmD. And what we did was collect 8 patients with Dravet's, with verified SCN1A mutations in Dravet's, and compared them to age and sex match controls. And they were not on a ketogenic diet, and this required just collection of 10 milliliters of blood samples from each of these patients. And we were able to transform these, the blood cells, the B cells from the blood into immortalized lymphoblast cell lines, and Anna was able to conduct some metabolic function assays and also ask what was wrong with the cell metabolism at the level of the mitochondria. And what she did, this is just a graph that shows you what a typical mitochondrial function test would look like in cell models. And you see oxygen consumption in the presence of activators and inhibitors of the electron transport chain. And a typical graph should look like this. And what we see on the right in the controls and Dravet syndrome patients is that there is a decline in energy consumption, mitochondrial energetics, or oxygen consumption in the Dravet group compared to the control group. And if you compare the different parameters that you can measure, we see a decrease in energy production and maximal as well as spare respiratory capacity. What Anna also asked is which of the electron transport chain complexes was impaired or dysfunctional, and what she found was it was complex 1, which was most dysfunctional, followed by complex 2. So this gives us an idea that this kind of is a mitochondrial disorder that looks a lot like conditions like Parkinson's, in which there are these complex 1 dysfunctional. The next question was in a mouse model, and this is work done by Hydrin Poshka's lab, in which she took a heterozygous humanized Dravet syndrome mouse model and did a metabolomic analysis. And here is difficult to kind of see all the changes, but this is at synaptic level in metabolism, and all the red areas are where there's increases or decreases in metabolism, and the blue are where there's increases. And what they found was there was an alteration in glucose intermediates and an increase in glycogenolysis and glycolysis that occurred in these mouse regions. And then we've also gone on to look at the zebrafish model of Dravet syndrome. So the zebrafish model has been developed and used by Dr. Baraban's lab at UCSF, and we have collaborated with him to use this model to understand metabolic defects. So zebrafish have duplicate alleles of this SCN1A. So in this model, one of them is a natural mutant, and so it's dysfunctional, and so we have a heterozygous kind of condition that resembles human Dravet syndrome in its ability to produce spontaneous seizures, temperature-related seizures, and they are resistant to certain medications that patients are, and responsive to others. They also respond to certain ketogenic diets and show some of the other phenotypes of mouse models and humans. So we asked in the lab, is there any metabolic dysfunction in this model? And these zebrafish larvae were placed in this metabolic chamber, if you will, and measured for their glucose metabolism. And this set of graphs shows you that baseline glycolysis was decreased like a hypometabolic state. And there were also genes that resulted in proteins that would increase glucose expression, such as those in the gluconeogenesis pathway that were downregulated in this model. And then when you look at their respiration at the mitochondrial level, and this is a busy graph, but what it shows you is a lot of the parameters that are important for oxygen consumption at mitochondria were also decreased, such as the maximal respiration, proton leak, and spare respiratory capacity. So these zebrafish had a decrease in their mitochondrial respiration. So this really kind of validates some of the other models and the human condition in terms of glucose hypometabolism as well as mitochondrial respiration. This is a slide that I obtained from Dr. Chad Fraser at Eastern Tennessee University, and he is studying SUDEP and the SCN1 mutant mice. So this is a mouse model of Dravet syndrome again, and he's interested in cardiac function. And so he's really looked at these mice, and what he's found is something really interesting. On the leftmost graph, you can see that, and this is a phenomenon that happens more in the male offsprings than the female offspring. So there's a sex-dependent effect, and there was a decrease in mitochondrial function. There was an overutilization of complex 2 that resulted from decreased mitochondrial respiration, and purple bars are the Dravet syndrome mice, and the green ones are the wild-type normal mice. There was also an increase in mitochondrial reactive oxygen species production in the cardiac tissue, an increase in the deficits in the Dravet syndrome mice that resulted from a sympathetic tone to the heart. And so this could predispose these mice to SUDEP, and mitochondrial reactive oxygen species are a prime mechanism for arrhythmias in the heart. So Chad's lab has very nicely shown that the mitochondrial function is damaged in these cardiac tissues. So this gives us some evidence for comorbidities of Dravet's as well, not just the seizures themselves and the disease states. So this slide is actually adapted from Karen Borges' lab that really nicely summarized the metabolic impairment in Dravet syndrome from 3 different conditions, from the human cases, the mouse model, as well as the zebrafish model in our lab. And what it shows you is a common theme of glucose hypometabolism and mitochondrial abnormalities that occur in these models. In our lab, we also showed the lymphoblast cell lines that have mitochondrial defects, and then Chad's lab has shown these defects in the cardiac tissue. So we see hypometabolism and complex 1-2 defects. So next I want to take you to the acquired epilepsy. And as you know, the process of the acquired temporal lobe epilepsy, for example, it begins with some sort of traumatic injury or hypoxic, ischemic, infectious injury to the brain that kind of results and culminates into a long-lasting hyperexcitability and spontaneous seizures. And the acute and latent stages of this condition are really complex, and they involve a lot of different processes, signaling mechanisms, metabolic alterations, inflammation. But you can really stage this chronic acquired epilepsy into 3 major areas, into the acute, latent, and chronic phase. And so our goal has always been to kind of understand metabolism and redox regulation in these phases. But I want to start with the human condition and hypometabolism that has been observed in human temporal lobe epilepsy. So this has been commonly observed that the PET imaging studies revealed a decrease in glucose metabolism in the epileptogenic temporal lobe during the interictal period, and this happens regardless of visible MRI damage. There's progression, and the outcomes can be pretty negative, resulting in cognitive decline and persistent seizures. Often used as a biomarker for the epileptogenic regions, and has a prognostic value when it comes to surgery for epilepsy. However, the causes of this hypometabolism are not well understood so that we can address this. In our lab, this is a rat study. We had conducted the similar experiment that I just started with staging the different phases of epilepsy using the canic acid rat model. And we start out by delivering status epilepticus to the rat and then understand the metabolic alterations that occur for periods of 6 to 12 weeks. And this is an experiment that was done by Dr. Shane Rowley, who was a graduate student at the time. And what Shane observed was similar to what the earlier graphs I showed you with the respiration, that he observed a decrease during the acute stages after status epilepticus, which recovered and then were reignited in the chronic phases of epilepsy, where you see a decrease in mitochondrial respiration. So the question that comes to mind is, what are the complexes in this acquired stage of epilepsy that are affected? And the human condition has shown that, again, mitochondrial complex 1 deficiency does occur in mesial temporal lobe epilepsy in the epileptic foci. And this was work done early in 2001 by Wolfram Kunz and Christian Elger. And it really showed very elegantly not only the complex 1 defect, but a decrease in N-acetyl aspartate, which is a mitochondrial kind of a biomarker, if you will. In our lab, we asked if the animal model, the canic acid model, showed similar complex 1 defects, and this is work by a graduate student, Kristen Ryan, who showed that mitochondrial complex 1 activity was decreased in the canic acid model shortly after status epilepticus, returned to control values in the latent period, and then was decreased again chronically. And she also found the mechanism of this dysfunction was due to a single alteration by an oxidative post-translational modification of one of the subunits. So complex 1 is a very large complex composed of over 50 subunits, and one of the hotspots of this complex was modified, which explained the transfer of NADH to the complex and a decline in the activity. So we have somewhat of a metabolic explanation of this complex 1 defect from animal studies. So this kind of leads me to mitochondrial complex 1 and other mitochondrial deficits, and to understand, is there a significance, and could this potentially contribute to the hypometabolic state that we see? So this is a sort of a picture that we can combine with all of other data that we have. So under normal circumstances, mitochondrial complex 1 works by having NADH transferred to it, and the electrons from here are transferred to ubiquinone, which is next to the complex, and protons are pumped efficiently, and downstream ATP is produced. However, when you have a defect in complex 1, there's an increase in NADH, which backs up a decline in NAD+, which is this redox partner of NADH, a decline in ATP production, and a consequent increase in mitochondrial superoxide or reactive oxygen species production. Could this mitochondrial defect then lead to its chronic hypometabolism? And this happens because this decline in NAD plus is a kind of a stimulus. As I mentioned earlier, when there's increase in glycolysis, there's an increase in release of lactate, and lactate is produced, and LDH, which is the lactate dehydrogenase, is activated to really produce this sustained lactate for NAD plus production. We also need some ways to increase the glycolytic pathway or glycolysis, and what has been shown is that the LDH increase is dependent on two pathways, signaling pathways that involves AMP kinase, which is like the energy charge that's lost, and HIF1 alpha. Both of these processes are able to increase the transporters for glucose and really give the tissue a glycolytic phenotype. So this way we could have a reprogramming of metabolism in which the NADH that is increased could lead to a LDH-mediated lactate production, and the mitochondrial alterations leading to a hypometabolic state. So what happens is the brain is really kind of hunkering down and not utilizing much of its energy, but going into kind of a survival mode and conserving energy, which is what hypometabolism kind of can be translated into words would be. So this is one hypothesis about how you could get a chronic hypometabolism in an epileptic brain. Could it be genetic or acquired epilepsy? And lastly, I want to talk a little bit about the therapeutic approach, starting with pharmacological approaches. So I mentioned that when you have these defects, you could increase steady state levels of reactive oxygen species, and mitochondria are prone to do this because they are utilizing oxygen for its energy production, and any slip of electrons can result in increased steady state superoxide and hydrogen peroxide production. In the epileptic brain, in acquired models, our lab and others have shown that there are two major sources of reactive oxygen species. That is the NADPH oxidase that are membrane bound at the cytoplasm, as well as the mitochondria. And really, we want to understand these are more damaging species when they're chronically produced. So you really want to try to therapeutically modulate these. The other phenomenon that has been observed from human conditions to animal models in our lab was, some of the initial work in our lab was done on this is glutathione depletion. That is a small molecule antioxidant, and it really is a readout for what the redox state of the tissue or the cell is. So what we see in the canic acid model of epilepsy, which is shown here, is kind of a sustained, a persistent decline in this redox couple, which tells you that it is a much more oxidized state in the long term. So can we intervene? And there's two different ways to intervene if you want. There's many ways you'd find things on in the internet of how you want to take antioxidants, lather them on yourself, all of these different things. But for therapeutic purposes, there's either direct antioxidants that can directly scavenge, and hopefully you have a catalytic one that can give you back the original compound, and you don't have to take it in large amounts like a vitamin. Or you can have indirect antioxidants that may be able to really upregulate your body's own antioxidant defense or are precursors to natural antioxidants. So I'm going to show you an example of each of these. So this is an experiment that was done by Dr. Jen Pearson-Smith, who was at the time she was a graduate student in the lab, and she asked, does a small molecule catalytic antioxidant modify epilepsy or cognitive dysfunction? And she used this porphyrin-based compound that is able to penetrate the blood-brain barrier, and it can perform 1 or 2 electron transfers, and it's more like an enzyme, so it's catalytic in its function. And what she did was administered the compound following status epilepticus, so it's a much more translational model, and she did a short-term treatment, so it's just a 2-day treatment, and then she weaned off the rats and then let them grow up a little longer, and she assessed chronic epilepsy and cognitive function. And what she found was that the reactive oxygen species production was increased. So this is shown as glutathione redox couple, which is depleted and prevented by this compound. In the yellow bar, you can see the prevention. The mitochondrial respiration was also rescued. Neuronal loss was rescued by the compound. And what she observed with this short-term treatment was that the cognitive function was improved, but epilepsy was not. So the next experiment I want to show you is that done in Anna Maria Vizzani's lab, and this was in collaboration with our lab and many others. What they did was they combined 2 indirect antioxidants. One is a precursor of glutathione, and the other is sulforaphane, which is a compound that can activate a signaling pathway that increases the body's natural antioxidant enzymes. And this was done in a subchronic manner, so it was done for 2 weeks continuously, and then they measured cognitive dysfunction and epilepsy development. And the results are shown in this graph. So chronic administration of this sulforaphane and N-acetylcysteine combined was able to delay the onset of seizures, as shown in the top left, And so the treated animals have a delayed onset. They had a prevention of spontaneous recurrent seizures. They also showed a reduction in the burden of seizures, and importantly, they also showed a cognitive improvement with this combination. So inhibition of oxidative stress in a chronic manner was able to improve the outcomes of both epilepsy and its comorbidity, which is cognitive decline in this case. And so next, I want to kind of shift to dietary therapies, which have been really important in our field. Our field has led to historically to the recognition that ketogenic diets, metabolic therapy, have very strong anti-seizure properties in many different disorders. And so ketogenic diets, as you know, can be come in a classical form or different varieties, but they are really a difficult therapy for patients to sustain and adhere to. And so we have, as a field, been really looking at kind of the holy grail of the ketogenic diet, trying to reverse engineer it to understand what targets could be employed to turn into pills or smaller oils, or what other types of conditions that we could really treat. So it's really been a lot of work in the field, and a lot of people here have contributed to this field. And we've shown together that many pathways, including adenosine, the mTOR pathway, AMP kinase, PPARs, and sirtuins and HDACs are all part of this. Other large pathways such as reactive oxygen species and inflammation also contribute to some of the protective effects of ketogenic diets. So I just want to kind of bring back what the different targets we could now learn from. Based on what I've told you, you may be able to predict very easily what targets would be important for seizure control or for cognitive improvement in the epileptic brain if you were to look at metabolism-based approaches. So as I showed, this is a busy slide because it kind of encompasses some of the mechanisms that are really protected by the ketogenic diets, but also mechanisms that are uncovered by understanding metabolic impairment in genetic and acquired epilepsies. So we see that you could block acutely glycolysis or glucose utilization. LDH inhibition has shown to be protective against seizures. Chronically, I've been reading some papers about GLP-1 chronic use in people with epilepsy that may have some improvements in their outcomes. So that's going to be an open question for the field. It also decreasing reactive oxygen species production or scavenging them would be another way to improve outcomes. Neuroinflammation is a subject, a large subject, which I did not really go into, but that has a strong crosstalk with metabolism and reactive oxygen species production, and that's a strong target. That's also something that ketogenic diets improve. And then neurotransmission itself is very important because many of our neurotransmitters are synthesized partly in mitochondria. So mitochondrial dysfunction is really not a favorable outcome for neurotransmission, which we depend very strongly on for therapeutic outcomes in epilepsy. And then lastly, there's going to be a lot of studies that have been coming out and will come out on the gut microbiome and how metabolites in the gut would impact the brain's excitability. So with that, I want to summarize. I want to tell you that what we learned is metabolic dysfunction does occur in these prototypical epilepsies, both genetic and acquired epilepsy. Metabolic reprogramming and mitochondrial respiration defects, such as in complex 1, may contribute to the hypometabolic phenotype that we see. And metabolism redox-based therapeutic approach could reduce the burden and comorbidities of seizures. And I really think that ketogenic diet research could provide some new therapeutic avenues for the field. So I want to just circle back to the initial question that I posed, which is there a metabolic paradigm for epilepsy and its comorbidities? So the current evidence does suggest that both in certain genetic epilepsies as well as certain acquired conditions, in human to experimental models, there is some glucose hypometabolism overall. Mitochondrial dysfunction has been observed, oxidative stress has been observed, and therapeutic response to metabolism-based approaches has been observed. So a metabolic paradigm for epilepsy and its comorbidities, I would argue, is rapidly evolving. So what can you do from the clinical side? Well, be on the lookout for abnormalities and be open-minded, I guess, about this sort of research and area that could underlie both the genetic as well as acquired epilepsy. And also look for metabolism-based therapies that may be in the pipeline to treat patients with epilepsy. And so I have a very big slide for acknowledgements again. I want to thank my funding, for NIH continuous funding for these many years. And some of the work that I mentioned was supported by the Dravet Syndrome Foundation and some of the other funding agencies like CURE. And so I'm very grateful for that. Some of the fellows were supported by some of the fellowships that we've provided. I'm very thankful to my collaborators and my AES colleagues for their help with the research. This is our CU Anschutz Dravet syndrome team that does basic and translational epilepsy. And then if you're wondering like who's going to do this work, I have been cloned. This was a Halloween 2025 experiment that my students, 5 of my students, they look fresher, younger, more energetic versions of me. And so you're going to have lots of Manishas out there. And with that, I want to thank you for your attention and for coming.
Thank you, Manisha, for setting the bar high for the rest of the meeting. Again, welcome to everybody to AES 2025, and I hope that you have a very successful meeting as well. Just a quick reminder that for those of you who have benefited from NINDS funding, that we will be taking a picture up here to celebrate what NINDS has done for AES and epilepsy research over the years. So again, thank you for joining us this afternoon.